Hello everyone, very good evening. Good evening students. How are you all? Welcome back. Welcome back to the continuation series of photosynthesis, that is part 2. Good evening everyone. Please confirm everyone if I'm audible and visible. Please come on, type yes if I'm audible and visible to everyone. So if yes, so hey, this is Riya Kilwani. This time I will be dealing with the revision of all the important topics that is there in NEET Biology. So if any new student has joined here, so for your information, let me tell you, we have come up with an exclusive Nail the NEET Revision Series. So we will be going to live for 15 days and today it is the 4th day of this series. So if you are new here, if you have any queries related to it or you wanted to know more about the series, you can just join our Telegram group, the link that is given in the description below. Or you can also call us on a toll-free number that is displayed on our screen. If not, you can also write a mail to us at info@biotecnika.org. If you'll write this, our technical team will revert you back immediately. So we will be waiting for 1 or 2 minutes and then we'll start with the session. Good evening, Smita. Good evening, Selva. Good evening, everyone. Srivani, very good evening. So we'll wait for 1 to 2 minutes. Till time, you can also share this link to all your friends so that more number of people will join and we can then start the session. Welcome back, everyone. So today we are going to finish this photosynthesis in higher plants. In previous class, that is yesterday's class, we have already covered half of the syllabus of photosynthesis. So now the remaining topics today we are going to cover in this session. So be tuned and connected till the last part of it so that you can just finish this topic in just one day. Good evening, Shivangi. कैसे हैं सभी लोग? ठीक है? So yes, so let us start the session. Let us start, guys. Very good, Smita. You're fine. Shall we start the session? Yes. So now we are going to start our session. So yesterday we have already covered what is the mechanism of photosynthesis, and in that we have covered what is cyclic and what is non-cyclic photophosphorylation, and we have also studied the difference between the two. But we have not covered yesterday what is chemiosmotic hypothesis. So this hypothesis is generally known for the generation of ATP or for ATP production. How ATP is produced in the case of non-cyclic photophosphorylation. So we'll understand how this hypothesis, it was previously given and after that it turned into a theory. So this theory is given by Peter Michel in 1961. Again, you can get a question in NEET examination for the name of the scientist. So it was given in 1961 by Peter Michel. So let us understand what is this chemiosmotic theory. So if you recall yesterday's lecture, we discussed about non-cyclic photophosphorylation. Non-cyclic photophosphorylation. So in this photophosphorylation, which is taking place in the thylakoid. So we are drawing here thylakoid, the structure of thylakoid. So what is this? This is thylakoid membrane. Thylakoid membrane. And outside this, it has stroma because these structures are present in the chloroplast. So obviously the stroma will be present outside the thylakoid. So what is this? This is a viscous fluid called stroma. All right. So this is thylakoid membrane and inside is a lumen. So recall the diagram of non-cyclic photophosphorylation. Firstly, pH2 was there, which was just above the membrane. And below this pH2, there is a photolysis of water happening. Now, what is this photolysis was doing? This was generating 4 hydrogen protons, 4 electron plus 1 oxygen molecule that is released back to the stroma. But if you see, 4 protons are generated here. Now, एक water molecule तो नहीं टूटता. Many water molecules are there that are getting splitted because photosynthesis is a continuous process. ऐसा नहीं है कि it is happening just for the one time. So just recall कि अगर इतना H plus proton will be increased in the lumen, the pH will go down. The pH of the lumen will become acidic. So what happens if pH2 is sending the electron to primary electron acceptor? So what is the primary electron acceptor in case of pH second? It is pheophytane. So when this pheophytane sends the electron to the next electron acceptor, that is plastoquinone, it is getting reduced. This plastoquinone is getting reduced to PQH2. क्योंकि PQH2 is formed and this plastoquinone बोलता है कि I'm not just accepting the electron, I also want a proton from the outside. So जब ये प्रोटॉन बाहर से एक्सेप्ट करता है तो यहाँ पे फिर प्रोटॉन की कमी हो जाती है. There is a lack of proton in the stroma. एक तो ल्यूमेन में प्रोटॉन वैसे ही ज्यादा बढ़ गया because of this photolysis. Again this PQ form, it is accepting the proton from the outside, which results in a very low pH in stroma. So what is happening here? This low pH, it is ultimately generating a electrochemical gradient. Now what is this electrochemical gradient? The difference between the two. किस चीज का difference है? pH, because of hydrogen ions or simply protons. Why it is generated? It is firstly generated because of the photolysis of water and second because of the plastoquinone, which is accepting the protons from the stroma. Now when this electron travels downhill, it sends the electron to plastocyanin and then ultimately to photosystem 1. Now what this photosystem 1 is doing? It is sending this electron to the ferredoxin, that is the primary electron acceptor in photosystem 1, and after that to NADP reductase. Now what is this doing? This NADP reductase, it is reducing NADP to NADPH2. Now again this is a reduction process. So what will happen here? If this reduction is occurring here, it will always require hydrogen ions. Now from where this hydrogen ion again coming? It is coming from stroma itself. So third responsibility किसने ली है कम करने की प्रोटॉन? It is only NADP reductase enzyme. So you can see तीन तरीके से स्ट्रोमा में H plus ions की कमी हुई. Firstly, this H plus ions, it is getting increased in the lumen of thylakoid because of the photolysis of water. Second, because of the proton carrier, that is a plastoquinone. And third, because of the reducing power generation, that is NADPH2 formation. Now what will happen here? If इतना ज्यादा प्रोटॉन अंदर है और इतना कम प्रोटॉन बाहर है, obviously there is a generation of electrochemical gradient. और electrochemical gradient generation होने के बाद there is a need to reach the equilibrium condition so that there should be an equivalence distribution of protons inside the lumen as well as in the stroma. So what will happen? Now there comes a ATP synthetase. Now this is a F0 particle and this is F1. Now what this particle does? F0, F1. F0 is known for the facilitated diffusion, whereas F1 is a ATP synthetase. Now what it will do? This F0 particle, it will accept all the protons that is there in the lumen. It will accept, and using this protons as an energy, this F1 particle, which is behaving as an ATP synthetase, it will convert ADP to form the ATP. Now it will do that till that time जब तक दोनों के बीच में protons will not become equal. So this F0 particle is acting as a channel for the protons because it is performing a facilitated diffusion of these protons. So I hope this diagram is clear to everyone. I'll just give you a quick recap of this diagram. So we have already studied in non-cyclic photophosphorylation that ATP is generated. So we are going to understand how this ATP is generated. It is generated by the help of chemiosmotic hypothesis. So this chemiosmotic hypothesis, firstly PS2 is acting. In this PS2, this proton is generated by the photolysis of water. And when this proton is further carried to plastoquinone, again it is a proton carrier that accepts the proton from the stroma. And if this proton is created in the lumen, and if the plastoquinone is accepting the protons from the stroma, it will ultimately result in the decrease in the proton gradient in the stroma. Because अब स्ट्रोमा में बहुत कम pH H plus है तो pH will be very high and lumen में pH is very low and ultimately this reducing power is also formed at the last जिसके कारण से proton gradient बहुत कम हो जाता है stroma. So there is a need to balance this proton between the thylakoid lumen and the stroma. So for this chemiosmotic hypothesis is running. So here F0 and F1 particle is acting or it is playing a key role. Here F0 particle is helping for a facilitated diffusion of these H plus protons, whereas F1 particle is a ATP synthetase, which is ultimately generating a chemical compound that is ATP. So let's come back to the slide and read this NCERT diagram, what it is saying. So firstly, light that is harvested by PS2, photolysis is taking place. It will result in the increase in the proton gradient inside the lumen. And again, plastoquinone, it is accepting the proton from the outside. Again, the proton gradient will be more in the lumen. And ultimately, this NADPH ka formation, it is again taking 2 electrons from the stroma, which will again reduce the pH in the lumen of thylakoid. So ultimately, F0 and F1 particle is acting, leading to the generation of ATP molecule. So I hope this process is clear to everyone. If yes, then type yes in the comments below. If you have any doubt, then type your queries related to it. I'll explain the doubt again. Okay, clear. Thank you, Anita. Okay, now we'll go to the questions. The first question that you have on the screen is chemiosmotic hypothesis that is given by Peter Michel proposes the mechanism of for the synthesis of ATP. Is it for the synthesis of NADH, synthesis of FADH2, or for the synthesis of NADPH? Shivani is saying option 1 for the synthesis of ATP. All right, well done, Shivani. The correct answer is ATP because chemiosmotic hypothesis, it was given by Peter Michel to just derive the production of ATP, how ATP is synthesized in case of non-cyclic photophosphorylation. Now let's come to the question number 2. The chemiosmotic coupling hypothesis of Oxford proposes that ATP is formed because ATP क्यों form हो रहा है? A proton gradient is formed, or a change in permeability, या high energy bonds are formed, or ADP is pumped out into the intermembrane space. What is the right answer for this question? Anita is saying, Srivani, well done, Smita, A. So yes, a proton gradient is formed across the inner membrane. Proton gradient कैसे बन रहा था? It is just because of the electrochemical gradient that is caused due to the difference in the distribution of protons inside the lumen as well as in the stroma. So let us read more options. There is a change in the permeability of inner mitochondrial membrane. First of all, हमने chemiosmotic hypothesis हमने कहाँ पढ़ लिया कि वो mitochondria में होता है. It is always taking place in the chloroplast. So this option is going wrong itself. Third option है high energy bonds are formed in the mitochondrial protein. Again, this is wrong because chemiosmotic hypothesis, it is taking place in a structure called chloroplast. ADP is pumped out of the matrix into the intermembrane space. हमने कब पढ़ा कि ADP बाहर आता है? जो ADP already it is present in stroma, it is converted to ATP. So ultimately, the first option is right answer. Now, third question, chemiosmotic theory of ATP synthesis in the chloroplast and the mitochondria. It is based on membrane potential, accumulation of K plus ions, accumulation of Na plus ion, or proton gradient. इसलिए होता है. So it is because of the proton gradient, again H plus ions. So I hope the questions are clear to everyone. So yesterday we have already covered what is photosynthesis, its mechanism, light independent, light dependent. So in light dependent reactions, we have already covered cyclic and non-cyclic. And also we have seen the chemiosmotic hypothesis that is related to non-cyclic photophosphorylation. Now today we are going to deal about what is light independent reaction. Now let's come to what is light independent reaction. So light independent reaction is also called as dark reaction. A dark reaction doesn't mean it is always occurring in the absence of light. It can occur in the presence or absence of light. So here dark reaction is a misnomer. Why? क्योंकि dark से ऐसा समझ में आता है it is always occurring in the absence of light. But ऐसा नहीं है. It can occur in the presence or absence, but it requires molecules that are formed in light reaction. So it is dependent on the assimilatory molecules that are already formed in light reactions. Now, what are these assimilatory molecules? We have already studied yesterday. It is ATP as well as NADPH2. So where it is taking place? It is taking place in the stroma. It is also called as biosynthetic phase because here occurs the production of glucose, which will ultimately be converted to starch. So इसका discovery कैसे हुआ? Dark reaction का. So this dark reaction, it was discovered by Melvin Kelvin. So to give honor to the scientist, this dark reaction is also known as Kelvin cycle. Now, during the World War II, बहुत बातें हो रही थी. Negatives about radioactive isotope कि ये हमारे किसी काम के नहीं हैं. तो seeing all these talks, तो मेल्विन कैल्विन ने क्या किया? एक 14 heavy isotope कार्बन का prepare किया. And he used this heavy isotope of carbon in his experiment. So he used chlorella or Senedesmus as a green algae for his experiment. So what this green algae is doing? It is basically performing the photosynthesis. तो ये उनके लिए experimental model का काम किया. What? Chlorella and Senedesmus. You can have a question from here. So just be focused what I'm explaining. What all points I'm writing here, it's very important for the NEET exam point of view. So उनने देखा कि ये photosynthesis कर रहा है. Why this Melvin Kelvin he is taking chlorella and Senedesmus? Because it is clearly be taken in a test tube. It is a very small algae. So it can perform photosynthesis and can be taken in a test tube as well. So how this photosynthesis is taking place with the help of Calvin cycle, we'll see this. तो क्या किया, इनने एक कार्बन को मार्क कर दिया, रेडियोएक्टिव लेबल, and once he marked this carbon, and जब Calvin cycle चला तो he was getting 3 carbon stable compound as a product. तो उनने सोचा दिस कंपाउंड वी आर गेटिंग मे बी 1 कार्बन इसमें लेबल हुआ होगा and 2 carbon are coming from other source. So what is this source? So he was finding this source कि जो कार्बन लेबल होके 3 कार्बन प्रोडक्ट के फॉर्म में आ रहा है, but what is this 2 कार्बन कंपाउंड that is joining with this 1 कार्बन. फिर उन्होंने सोचा no, this is wrong, something else is there which is accepting this carbon dioxide. Now let's see who is accepting this carbon dioxide in the atmosphere. So you have 3 phases in Calvin cycle. The first is carboxylation. The second is reduction, and the third is regeneration. Now what happens in the carboxylation? RUBP comes and it accepts CO2. When RUBP accepts CO2, that means it is known as carboxylation. Now RUBP is acting as a carbon acceptor here. What is this RUBP? It is ribulose 1,5 bisphosphate, which is known as the carbon acceptor. Important point. पहला पॉइंट हमने निकाला नीट के लिए कि जो फर्स्ट कार्बन एक्सेप्टर है इन द कैल्विन साइकिल, इट इज RUBP. Now it is taking place in the presence of enzyme which is called as ribisco. Now what is this ribisco? Ribulose 1,5 bisphosphate carboxylase oxygenase. क्योंकि ये कार्बोक्सिलेज है, that is why it is accepting carbon dioxide from the atmosphere. So what happens? It will lead to the formation of 2 molecules of 3-phosphoglycerate or phosphoglyceric acid. Now this phosphoglyceric acid is a 3 carbon compound. So when 3 into 2 combines, it will gives to 6 carbon. तो देखते हैं 6 कार्बन था स्टार्टिंग में कि नहीं. 1 carbon we are getting from carbon dioxide and RUBP is a 5 carbon compound. So RUBP 5 carbon plus 1 6 carbon. So ultimately we are getting 6 carbon at the end. So how we are getting 3 carbon of phosphoglyceric acid into 2. Now what happens? This is called as carboxylation. So our carboxylation is completed here. Now it's a time for reduction. This reduction process, it takes ATP from the outside as the energy source and convert to ADP. It also uses NAD positive and converts to NADH2. So here you can see H2 and converts to NAD positive. So here you can see ATP is used. Here you can see ATP is produced as well as NADH2 is also used. So it is converted to phosphoglyceraldehyde. Glyceric acid, it is converted to aldehyde by the reduction process. So again 2 molecules of phosphoglyceraldehyde will be formed. So this is reduction. So next comes regeneration. So in this regeneration process, this is again 6 carbon. How? Because 2 molecules of phosphoglyceraldehyde, it will gives to 6 carbon. Now 1 carbon form here, it is fixed in the form of glucose. And we have 5 carbon remaining which will be regenerated in the form of RUBP again. So firstly carboxylation is taking place, then reduction, and then again regeneration. This regeneration again requires one ATP molecule. So this is for one molecule of carbon. But in glucose we have 6 carbon. तो जब एक कार्बन के लिए एक केल्विन साइकिल चल रहा है, then 6 कार्बन के लिए 6 Kelvin cycle will run. For 1 Kelvin cycle, 1 CO2 is accepted. So for 6 molecules of carbon or for 1 glucose molecule, 6 Kelvin cycle will be run. इतना समझ में आया? Now we'll understand how this is happening. So what is dark reaction? The pathway by which all photosynthetic organism fixes carbon dioxide and converts it to carbohydrate, that is glucose. It is known as dark reaction, biosynthetic phase or Calvin cycle. This dark reaction, it is not happening in dark but is dependent upon the product of light reaction of photosynthesis, that is NADPH as well as ATP. Where it is occurring in the stroma. First carbon acceptor is RUBP here. First stable product that is formed is 3-phosphoglyceric acid. And that is why this cycle is known as C3 cycle. Because the first stable product that is formed is a 3-carbon compound, that is phosphoglyceric acid. Now what is the enzyme that is used to catalyze this process? It is Rubisco, that is RUBP carboxylase oxygenase. It is the most abundant enzyme on earth. Again, you will have a question from here, important point. Where it is occurring, what is the acceptor, what is the stable compound, which enzyme is catalyzing this process, where it is occurring in the mesophyll cells of the leaves. Presence, it is occurring in monocots or dicots? In both monocots as well as dicots. What is the end product? At the end, we'll get glucose molecule that will be stored in the form of starch in amyloplast, or if it is translocating in phloem, it will translocate in the form of sucrose. So glucose will form starch or sucrose depending upon the requirement. So you can just see it is the NCRT diagram that is given here. Ribulose 1,5-bisphosphate, that is RUBP. It is accepting one carbon dioxide molecule from outside. Carboxylation is happening place. It will lead to the formation of 2 molecules of 3-phosphoglycerate, that is a 3-carbon stable compound. Now again reduction will take place. It will utilize ATP and NADPH as an energy source, and it will release ADP, inorganic phosphate, and NADP. So for one phosphoglyceric acid, 1 ATP will be utilized. But here 2 molecules of phosphoglyceric acid is forming. That means 2 ATP will be utilized as well as 2 NADPH. And triose phosphate will be formed and 1 glucose molecule will be released as a glucose molecule. So it will fix till 6 carbon dioxide molecule is released. It will fix 1 glucose. And ultimately this triose phosphate, whatever carbon compound is left, it will again lead to the regeneration of RUBP. And again this regeneration, it requires 1 ATP molecule. So we have seen in fixing 1 molecule of carbon, it is requiring 3 ATP. And how many NADPH are required? 2 NADPH required in the reduction phase and 2 ATP in the reduction as well as 1 ATP in regeneration phase. So these we have already covered phases of Calvin cycle. First is the carboxylation of RUBP, in which RUBP accepts carbon dioxide from outside and fixes the carbon in the form of 3-phosphoglyceric acid, which is catalyzed by an enzyme called Ribisco, which is one of the most abundant enzyme on earth. Now the second step over here is reduction, which requires 2 molecule of ATP and 2 molecule of NADPH to reduce this 3-phosphoglyceric acid into phosphoglyceraldehyde. Now this phosphoglyceraldehyde, it will release 1 carbon to fix into the form of glucose, and rest of the carbon, it will be regenerated in the form of carbon acceptor, that is RUBP. So the third step is RUBP. So regeneration of carbon acceptor molecule, which is the crucial in the cell cycle. So total Kelvin cycle is 6. How 6? Because if one Kelvin cycle will run, it will only fix one carbon molecule. But you have to fix 6 carbon to generate one molecule of glucose, because glucose contains 6 carbons in it. So total of 6 Kelvin cycles are required to generate one molecule of glucose. So 6 carbons are fixed to make one molecule of glucose. So for one glucose molecule, 18 ATP. How? Because in fixing one carbon molecule, it was requiring 3 ATP as well as 2 NADPH. So what will be the energy equivalence for the whole Kelvin cycle of a one molecule of glucose? Just multiply it by 6. So 3 into 6 is 18 ATP, or 12 NADPH is required in a process. So to fix one complete molecule of glucose, it is requiring 18 ATP and 12 NADPH. So this is very important. Just note down in your copy. So about 85% of the plant species are tree plants. So what are the examples of tree plants? You may get question from here. So be focused. It is barley, rice, oat, wheat, that is cereals, groundnut, sugar beet, cotton, tobacco, soybean, spinach, and trees. So we are taking 6 molecules of carbon dioxide. We are taking 18 ATP. We are taking 12 NADPH, and out what is the final product? We are getting one molecule of glucose at the last. So one molecule of glucose, 18 ADP and 12 NADP positive. So this is for half molecule of glucose, how half molecule of glucose is formed, and this is for complete one molecule of glucose. So whatever I have explained, you can just refer to both these diagrams according to your comfortable. So now let's go to the questions. If you have understood these things, now practice the question. So now let us move to the questions. Yes, Srivani, it is RUBP carboxylase oxygenase, because this enzyme has an affinity for both carbon dioxide as well as oxygen. That is why it is called as carboxylase as well as oxygenase. So it is a capital C and capital O, that is representing carboxylase as well as oxygenase. So you have question for dark reaction. In C3 plants, the first stable product of photosynthesis that is formed during the dark reaction is it malic acid? Is it oxaloacetic acid, phosphoglyceric acid, or phosphoglyceraldehyde? Selva option 3. Okay. Smrita, Anita, Srivani. Okay, Smita is saying option number 3. So now let's see. RUBP, when it was accepting carbon dioxide from the atmosphere in the presence of this enzyme ribisco, it was catalyzing the product process and forming 2 molecules of phosphoglyceric acid, that is a 3 carbon compound, and that is why this cycle is known as 3 cycle. So what is the final answer? It is phosphoglyceric acid, that is PGA. You can also call it as phosphoglycerate. तो अगर ऑप्शन में phosphoglycerate होता तो अगेन यही राइट आंसर होता. So if you write 8 or ic acid, it is the same thing. You can only write 8 or you can also write ic acid, glyceric acid. So now let's move to the next question. Dark reactions of photosynthesis, it occurs in grana, stromal lamellae membranes, photosynthetic lamella, or periprasteral space. Srivani, we have to just answer what is a stable product that is formed in the dark reaction. So the stable product that is formed is phosphoglyceric acid. This is the next reaction that is happening and it is leading to the formation of aldehyde. So the first stable compound that is formed is PGA, not aldehyde, because aldehyde is the second compound which is formed in the reduction process after phosphoglyceric acid. So the right answer over here is phosphoglyceric acid. अगर ये होता कि रिडक्शन में क्या बन रहा है तो the right answer would be option 4. But अभी ऐसा नहीं है. It is asking for the first stable product. First. So it is always phosphoglyceric acid. So yes, so the right answer for this question is stromal lamellae membranes, because dark reaction, they are taking place in the stroma. So I hope this is clear to everyone. So for assimilation of one carbon dioxide molecule, the energy that is required in the form of ATP and NADPH2 are ATP पहले बताना है, then NADPH2. How many ATP are utilized to fix one carbon dioxide molecule? Okay, Srivani. How many ATP are utilized to fix one carbon dioxide molecule? It is 3 ATP. 2 in the reduction process as well as 1 in the regeneration of RUBP. Whereas NADPH, they are utilized only in the reduction process and where 2 molecules of NADPH are utilized. So the right answer for this question is 3 ATP as well as 2 NADPH2 molecule. So the third option is the right answer. So for the synthesis of one carbon dioxide molecule, the Kelvin cycle operates for. Kelvin cycle कितनी बार run होगा for the synthesis of one carbon dioxide molecule? Make it as one glucose molecule. How many Kelvin cycles are required for the synthesis of one molecule of glucose? Very good, Smita, Silva. So for one glucose, how many Kelvin cycles are required? So for one carbon dioxide, one cycle was running and it was fixing one carbon as a product. So for 6 carbon dioxide to form one molecule of glucose, it will always require 1 into 6, that is 6 cycles. So the right answer for this question is 6 times. So the right answer is option number 3. Yes, good. Smita, Srivani, you all are right. Very good. So now let us see the differences between light reaction and dark reaction. So yesterday we have already covered this light reaction part and now we have covered the dark reaction. So now let us see what are the differences between the two. So light reaction, it is a light dependent process, whereas dark reaction, it doesn't require light. It doesn't require, but it can take place in the presence or well as absence of light. It involves photosystem 1 and 2. आपने आज देखा, is this process requiring photosystem? Never. बिल्कुल भी इसको photosystem की जरूरत नहीं था. Photolysis of water taking place and O2 is liberated. Here photolysis is not at all taking place and carbon dioxide is absorbed. How it was absorbed? By carbon acceptor, that is RUBP. It occurs within the grana of the chloroplast. This light reaction, it is taking place inside the grana of the thylakoid and it is taking place within the stroma of the chloroplast. So grana and this is stroma. ATP and NADPH are produced and they are used to derive the dark reaction. Here, this assimilatory molecules are produced. This is the energy source and this is again a reducing power. All these reducing power and energy source are utilized to form glucose in the dark reactions. So I hope the differences between the two is clear to everyone. If not, so you can post your queries in the comments below. So now we'll start with the C4 cycle. What is C4 pathway? So I hope that was clear to everyone. So C4 cycle. And then we'll come to photorespiration. Now this C4 cycle, it was given by Hatch and Slack. These scientists, they discovered this C4 pathway. And to give the honor to these scientists, this pathway is also known as Hatch-Slack pathway. Now again, these scientists, they were looking for the generation of the 3-carbon stable compound, that is phosphoglyceric acid, that was formed in the C3 cycle. But उनने देखा कि यहाँ पे तो 3-carbon product बन ही नहीं रहा है. It is always forming the 4-carbon compound as a stable product. और वो क्या था? Oxaloacetic acid. So what are the plants in which C4 cycle is taking place? It is always mass. And the mass here represents for M for maize, A for amaranthus, S for sorghum, as well as sugarcane. You have a question from here from these examples, so just make it star mark. So maize, amaranthus, sorghum, as well as sugarcane. Now we'll see how this cycle is running place. So firstly, the PEP compound, that is phosphoenol pyruvate. Phosphoenol pyruvate. It is accepting this carbon dioxide from the atmosphere. So once it is accepting, it is forming 4-carbon compound, that is oxaloacetic acid, or as well as malic acid. So once these compounds are formed, these are transported to the next cell, that is bundle sheet cells. Before that, I'll take you to the Kranz anatomy. So these C4 plants, they have developed one kind of anatomy called Kranz anatomy. Now what is this Kranz representing here? Kranz means wreath, and wreath means ring-like structure. Now these Kranz anatomy, they have two types of cells. One is mesophyll cells and other is bundle sheet cells. This bundle sheet cells has a very thick covering outside, which is making impervious to this membrane to air, that is oxygen. So it always has rubisco enzyme, but it lacks Papkase enzyme. Whereas mesophyll cell, it has a very thin covering. Now how these are arranged? Suppose these are bundle sheet cell and it has a very thick covering. And outside this, mesophills are arranged in a ring-like structure, that is what called Kranz anatomy. And these cells have a very low intercellular space. So no intercellular space is present between these mesophyll cells. They are tightly packed. So this bundle sheet cell, this membrane, it is impervious to oxygen or air. So always inside this, rubisco enzyme is present. Why rubisco? Because if it is present outside, it can also behave as oxygenase. And if it behaves as oxygenase, it will start the process of photorespiration that we are going to deal in the next slide. So it should always behave as carboxylase. Why? Because it has to fix carbon dioxide from the atmosphere to synthesize glucose. But if it uses oxygen, it will lead to the wasteful process. Because this is impervious to oxygen, so rubisco is always present in the bundle sheet cells. Now, what is this mesophyll cells containing? It is only containing pappcase enzyme, but in turn it has no rubisco. So now let us go back to the diagram and understand. So what happens? Firstly, phosphoenol pyruvate. It comes. It accepts CO2 in the presence of pappcase enzyme. So this phosphoenol pyruvate, it is a carbon acceptor in the C4 pathway. So again a star mark. In case of C3, it was RUBP. But in case of C4, it is phosphoenol pyruvate. So once it accepts carbon dioxide, it forms the oxaloacetic acid. This oxaloacetic acid, it can be interconverted to malate, that is malic acid, or aspartate, that is aspartic acid. So all these process, it is taking place in the mesophyll cells. Why mesophyll? Because pappcase enzyme is always present in the cytoplasm of mesophyll cells. So if it is present in the cytoplasm, so does it require grana or stroma? Because rubisco is not present here. So rubisco, it is always present in the stroma of the chloroplast. तो हम ये मान सकते हैं कि mesophyll cells में stroma बहुत कम है because rubisco नहीं है, that is taking care for the C3, that is Calvin pathway. So it has very less stroma because rubisco is absent in the mesophyll cells, and instead of this it has pappcase enzyme. So this mesophyll cell has very less stroma, but it has more grana. But in case of bundle sheath cells, it has rubisco enzyme. But this rubisco enzyme, we know it is taking place in the Calvin cycle, that is C3 pathway. So for this, stroma should always be present because Calvin cycle, it is taking place in the stroma. So if there will be more stroma, so it will have less grana. तो समझ में आया? We have two types of chloroplast here, and these chloroplasts are known as dimorphic because in mesophyll cells more granule chloroplasts are present because rubisco is absent, so stroma is not present. Whereas in bundle sheath cell, it has a rubisco enzyme, but rubisco, it is taking care of the C3 pathway, and that C3 pathway, it is running in stroma, so it has less grana. So these two type of chloroplasts, it is found in the C4 plants, that is taking care of this C4 pathway. Now once this C4 compound, that is oxaloacetate, it form, it will lead to the formation of malic acid or aspartate. Why this double arrow is shown? Because it represents the process is interconvertible. It can either be malic acid or aspartic acid depending upon the condition, क्या surrounding में need है वही compound बनेगा. So this compound will now be transported to bundle sheath cell. Now recall bundle sheath cell में rubisco present है. So we are now coming to that. Malate है, malate is a 4 carbon compound. Aspartic acid भी 4 carbon. Oxaloacetic acid is also a 4 carbon compound. So this malate, it will be converted to 3 carbon compound, that is pyruvic acid. So where this one oxygen is going? Because we have told that malate is a 4 carbon compound, but 1 carbon, it is going to fix in the form of glucose. So 1 carbon dioxide molecule, it will enters to the Calvin cycle. Now what about the 3 carbon molecule? It will form the pyruvic acid, and the pyruvic acid that is formed here, it will again be transported back to the mesophyll cells. It will take the help of the ATP, and utilizing this ATP, it will form the phosphoenol pyruvate. Why this is forming phosphoenol pyruvate? Because this reaction is initiated with the help of PEP. तो बहुत जरूरी है इसको regenerate होना. So who is regenerating this PEP? Pyruvic acid. Where it is formed? It is formed in the bundle sheet cells. So how it is converted to phosphoenol pyruvate or phosphoenol pyruvic acid? With the help of the ATP. Where this ATP is utilized? This ATP is utilized in the mesophyll cells. From where this ATP is coming? This ATP is coming from the light reactions. तो फाइनली हमारा PP एक्सेप्टर जो कि कार्बन डाइऑक्साइड को एक्सेप्ट कर रहा है, इट इज फॉर्म्ड हियर. नाउ यू कैन सी C3 cycle यूटिलाइज हो रहा है, and in C3 cycle we already know 3 ATP are utilized and 2 NADPH. And here also you can see ATP is utilized. So 2 ATP are utilized here also, because 2 molecules of pyruvic acid, it is forming 2 molecules of phosphoenol pyruvate. So 2 molecules of ATP are again utilized in case of C4 pathway. So total this C4 pathway, it is utilizing 5 ATP and 2 NADPH. समझ में आया? Why I am writing 3 and 2? Because here you can see C3 cycle is also taking place in case of C4 plants. So C3 cycle तो हमेशा ही चलेगी. Whether it is C4 plants, extra C4 cycle will be there. So this C4 cycle, it is extra, it is utilizing 2 ATP. But C3 cycle में अभी हमने questions में भी देखा, it was 3 ATP as well as 2 NADPH. So total 5 ATP are utilized in the C4 pathway along with 2 NADPH2. तो total consumption कितना हुआ? 5 ATP and 2 NADPH for 1 molecule of carbon. And suppose if it has to form 6 molecule of carbon, that is 1 complete glucose. So again multiply it by 6. So 6 into 5 is 30 ATP as well as 12 NADPH molecules. So here you can see more ATP is utilized in comparison to C3 pathway. So now let's come back to the slides and read what is written here. To overcome photorespiration, we'll understand what is this. C4 plants adapt to C4 pathway. Now what are the examples of these C4 plants? It is maize, amaranthus, sorghum, sugarcane. The trick to remember this is mass. Mesophyll cells and bundle sheath cells are connected with the help of plasmodesmata. So how this transport is taking place? It is taking place with the help of a plasmodesmata, which is a connection between the mesophyll as well as the bundle sheath cells. So this bundle sheath cell, it contains a large number of chloroplasts. Mesophyll cell contain a normal types of chloroplasts. And in this type of chloroplast, mesophyll cells, it has more grana. So granule chloroplasts are present because it has less stroma because of the absence of rubisco enzyme. So it has two types of adaptation. It has a dimorphic chloroplast in mesophyll cells. Why dimorphic? Because granule as well as agranule chloroplast are present here, and also it has developed trans anatomy in the bundle sheath cells. So this diagram we have already discussed. This is from NCERT image, so you can refer it. So is this diagram or pathway clear to everyone? If this is clear, so we can proceed with our questions. So okay, Silva, clear? Smita, Srivani, everyone clear? So yes, so I hope this is clear to everyone. So now let us deal the questions for this Hatch and Slack pathway. So first question for this pathway is which one is a C4 plant? इसमें से कौन सा example है which is a C4 plant? Is this papaya, pea, पोटैटो और मेज. जस्ट रिकॉल द ट्रिक. अभी मैंने क्या बताया था फॉर C4 प्लांट. वेरी गुड बायोलॉजी अनित, सेल्वा, देवांशी. ओके श्रिवानी, द राइट आंसर फॉर दिस इज मेज. ट्रिक क्या बताया याद करने के लिए? मास. तो पहला ही जो अल्फाबेट है इट रिप्रेजेंट्स मेज प्लांट्स. सो C4 प्लांट का एक एग्जांपल है मेज. Second question, C4 acid that is formed in the mesophyll of C4 plant's leaf during photosynthesis is oxaloacetic acid, malic acid or aspartic acid, or is it pyruvic acid or succinic acid or fumaric acid? Yes guys, come up. What is the right answer for this question? C4 acid जो बनता है hatch and slack pathway में, what is it? It is oxaloacetic acid or is it pyruvic acid, succinic acid and fumaric acid? Very good Selva, Devanshi. Very good. The correct answer for this question is oxaloacetic acid. First of all, हमने succinic acid के लिए कहाँ पढ़ा? Fumaric acid we have not at all discussed. We have just discussed about pyruvic acid and oxaloacetic acid. And pyruvic acid तो कहीं पे भी stable compound form ही नहीं हो रहा था. It was phosphoglyceric acid and that too in C3 pathway. So the right answer for this question is option A, where oxaloacetic acid it is interconverted to malic or aspartic acid. So in the leaves of C4 plants, malic acid formation during CO2 fixation occurs in the cells of. Where is this malic acid formed? Recall the pathway. Malic acid सबसे पहले कहाँ form हुआ था? किस type of cells में? Is it the cells of bundle sheath, phloem, epidermis, mesophyll? Biology Anit, he is saying 1, Selva 4. Anyone? Selva 1, okay. So we know in C4 pathway we are having two cells. First is mesophyll and the second one is bundle sheath. The first stable compound that is formed in a C4 pathway is oxaloacetic acid or malic acid or aspartic acid, which is a 4-carbon compound. So this malic acid is formed in the cells of mesophyll or mesophyll cells that is transported with the help of plasmodesmata to bundle sheath cells, where it is reduced to phosphopyruvic acid or pyruvic acid and 1 carbon is fixed here in the form of glucose. So the right answer for this question is option 4, that is mesophyll cells. Yes, right Srivani. Now we'll come to photorespiration. What is this photorespiration? Now this photorespiration, if you divide this term, photo stands for light और respiration यहाँ पे वो respiration नहीं है जो हम normally देखते हैं, which produces energy. But in this photorespiration, no energy is produced. और क्या होता है? एक तो एनर्जी नहीं बनती, ऊपर से ऑक्सीजन इज यूज्ड बाय रुबिस्को. अगर ऑक्सीजन रुबिस्को यूज करने लग गया तो हु विल एक्सेप्ट दिस कार्बन डाइऑक्साइड फ्रॉम द एटमॉस्फेयर? अगर कार्बन डाइऑक्साइड एक्सेप्ट नहीं होगा तो हाउ इट विल फिक्स इन द फॉर्म ऑफ ग्लूकोज? तो आरयूबीपी को तो एक्चुअल में कार्बोक्सिलेज की तरह बिहेव करना चाहिए था, बट हियर आरयूबीपी इज बिहेविंग एज अ ऑक्सीजनेज. सो ऑक्सीजन इज यूज्ड और जितना भी कार्बन फिक्स हो चुका होता है दैट इज रिलीज्ड जितना भी C3 प्लांट ने अभी तक मेहनत किया था कार्बन डाइऑक्साइड को फिक्स करने के लिए, दिस फोटो रेस्पिरेशन इट इज रिलीजिंग वन कार्बन डाइऑक्साइड मॉलिक्यूल, एक एक करके ये पूरा कार्बन डाइऑक्साइड रिलीज कर देता है. तो actually it is wasting the hard work of C3 cycle. So can we call it as a wasteful process? Yes, because no energy is generated, ऑक्सीजन यूज हो रहा है, कोई मतलब का नहीं है, बिकॉज कार्बन डाइऑक्साइड इज नॉट एट ऑल फिक्स्ड, एंड जो कार्बन डाइऑक्साइड फिक्स्ड हुआ वो भी रिलीज हो जा रहा है लीव से, तो ग्लूकोज तो बचेगा ही नहीं इन लीव्स. सो दैट इज व्हाई इट इज कॉल्ड एज अ wasteful process. So where this process is taking place? It is taking place in chloroplast, peroxisome, as well as mitochondria. So you can just learn it as a CPM of physics, chemistry, mathematics. बस सीरीज यू हैव टू लर्न CPM. और कैसे लर्न कर सकते हैं, कौन तीन ऑर्गेनिक्स है? PCM 12th में सब्जेक्ट लेते हैं, 11th 12th में फिजिक्स, केमिस्ट्री, मैथमेटिक्स. So now let's see how this process is taking place. So firstly, RUBP आता है, that is behaving as an oxygenase here. If it will behave as an oxygenase, so it will always accept oxygen from the atmosphere. Why it is accepting oxygen? कार्बन डाइऑक्साइड क्यों एक्सेप्ट नहीं किया? बिकॉज एट दिस पॉइंट ऑफ टाइम वेरी हाई अमाउंट ऑफ और हाई कॉन्सेंट्रेशन ऑफ ऑक्सीजन इज प्रेजेंट इन द एटमॉस्फेयर. तो वो ढूंढने तो निकला था कार्बन डाइऑक्साइड, बट इतना ऑक्सीजन देख के वो उसके लिए फेवर कर गया और उसने क्या एक्सेप्ट कर लिया? ऑक्सीजन. नाउ इन दिस ऑक्सीजन देयर इज नो कार्बन मॉलिक्यूल or there is no carbon. So this 5 carbon compound, it will combine to oxygen and will lead to the formation of phosphoglyceric acid, that is a 3 carbon compound, and it will form one molecule of phosphoglycolate. So total 5 carbon and RUBP, so it will break into phosphoglyceric acid, that is a 3 carbon molecule, and one molecule of phosphoglycolate will be formed, which is a 2 carbon molecule. Now this phosphoglyceric acid, it will again enters to the Calvin cycle, but phosphoglycolate, it will retain in the chloroplast itself. अब ये phosphoglycolate, it is a glycolate that is combined with an inorganic phosphate. So what will happen? This phosphoglycolate, it will release one phosphate in the form of Pi, and it will cause the formation of glycolate. Now this glycolate, it is highly toxic to the cell. ये बहुत ज्यादा टॉक्सिसिटी प्रोवाइड करता है सेल्स को, तो इसको ज्यादा लंबे समय तक प्रेजेंट होना बिल्कुल नहीं चाहिए. सो इट अगेन एक्सेप्ट्स one molecule of oxygen from the atmosphere and it will lead to the formation of glyoxylate. And once this glyoxylate is formed, it is also leading to the formation of hydrogen peroxide. Now this hydrogen peroxide is again a stressing agent for the cell. Why? Because it is a free radical. What it does? अगर ये लंबे समय तक मेम्ब्रेन के पास रहेगा, it will damage all the membrane. So how C3 process will take place? How glucose will be formed? Because it is going to damage the membrane. So this hydrogen peroxide, it is formed in the peroxisome and it is degraded here itself. So what is the place where hydrogen peroxide is formed? It is peroxisome itself, and this peroxisome is taking care for the degradation of this molecule. So this is degraded here. Now this glyoxylate, it is converted to glycine, which is an amino acid. Now this amino acid is a 2 carbon compound. Now from here till here, 2 cycles of photorespiration will take place so as to form 4 carbon compound. क्योंकि अभी तक we have got only one molecule of glycine and that too is a 2 carbon compound, but we want 4 carbon molecule to continue the process. So for this half photorespiration will again run to form 2 molecules of glycine. So once these 2 molecules of glycine are formed at the last, we have 4 carbon in hand. Now this glycine, once it is repeated, the cycle is repeated, we have 2 carbons, 2 molecules and total 4 carbons. Now this glycine, it is transported to mitochondria. After this glycine is converted to serine, which is a 3 carbon. हमने बनाया 4 carbon, 3 carbon यहाँ हो गया. क्या हुआ? 1 carbon का waste. That is why it is called as a wasteful process, because what all C3 plants has done to fix 1 carbon molecule of 1 carbon in the form of glucose, it is wasted in the form of carbon dioxide in the photorespiration. So this carbon dioxide is wasted, and along with it, 1 ammonia gas is also released. Now where this ammonia gas is utilized, it was again fixed by plants in the form of nitrogen assimilation. And where we are studying this, in the form of nitrogen fixation. So sorry, there was a technical problem. We are back now. So it was released in the form of ammonia. So again यहाँ पे plants ने hard work किया गया था to fix nitrogen, जो कि हम पढ़ते हैं nitrogen fixation. So this whole process is a wasteful process. Now this serine that is formed is a 3 carbon compound. It is transported back to peroxisome, where it is converted to glycerate, and accepting 1 molecule of inorganic phosphate, this glycerate is converted to phosphoglyceric acid, and this phosphoglyceric acid will then enters into a Calvin cycle. So here again you can see when this glycerate is accepting 1 inorganic phosphate, again it is utilizing ATP. Again एक तो ATP बन नहीं रहा, CO2 is not getting fixed, ऊपर से ऑक्सीजन एक्सेप्ट किया जा रहा है, और एट लास्ट एनर्जी इज कंज्यूम्ड. कुछ बन नहीं रहा, सब यूज किया जा रहा है. So this process is called as a wasteful process, which we call it as photorespiration. So the work that was done by the photosynthesis, it is undone by the photorespiration. Why undone? Because the in the form of glucose, it is getting released back again in the atmosphere in the form of carbon dioxide. So we'll read the details for this process. So C3 plants have disadvantage that in hot dry condition their photosynthetic efficiency suffers. Why it is written hot dry condition? Because there is high concentration of oxygen in the atmosphere. That is why we are covering these plants, C3 plants, in a greenhouse, so that जितना भी यहाँ पे photosynthesis और respiration हो रहा है, they will release, or this carbon dioxide will be released in the atmosphere, and we can trap this carbon dioxide. तभी हम इसको एक ग्लास में कवर करते हैं, that is what called greenhouse for C3 plants, because of a process called photorespiration. Note, अब C3 C2 cycle है, ऐसा नहीं कि C2 plants में होगा, because C3 cycle is what it was taking place in C3 plants, C4 pathway in C4 plants, but C2 pathway it is again taking place in C3 plants, and C4 pathway it is taking place in C4 plus C3 plants. But C4 plants का example है mass that we have already covered. So it takes place in C3 plants, that is bean, cotton, rice, wheat, barley, that are cereals. So what are the conditions for this C2 cycle? Why it is taking place? Because CO2 is less in the atmosphere. That means oxygen is high in the atmosphere. That is why ribosco enzyme, it is acting as a oxygenase. So O2 is high. That means if O2 is high, it is directly proportional to temperature. If O2 is high in the atmosphere, that means temperature is also high. So high temperature where ribosco is acting as an oxygenase but not carboxylase. So I hope this photorespiration is clear to everyone. Why this is called a C2 cycle then? Because the first stable compound that is formed in this cycle is glycolate or phosphoglycolate. That is a 2-carbon compound. तो ये जिनका भी नाम दिया गया है on the basis of the stable product that is formed in this pathway. C3 pathway, 3-phosphoglyceric acid, that is a 3-carbon compound. C4 pathway, oxaloacetic acid, that is a 4-carbon compound. And C2 pathway, phosphoglycolate is formed, that is further converted to glycolate, that is a 2-carbon compound. So photosynthesis, CO2 is fixed and O2 is given out. But in photorespiration, this CO2 become unfixed and O2 is taken in, because here Rubisco acting as an oxygenase. So what are the organelles? We have already covered: chloroplast, peroxisome, mitochondria. PCM as a code. Physics, chemistry, maths. Rubisco का property, it is a chloroplast enzyme. It can work as a carboxylase, as an oxygenase both, but it depends on the surrounding what gas is present in more amount. This enzyme is used in C3 pathway as well as C2 cycle. It has only one side. अब ऐसा नहीं है, if Rubisco is an enzyme, so it has two sides. No, it always has one side that has a similar affinity for carboxylase and oxygenase. It depends on the atmosphere which gas is present. If CO2 more it is present, it will accept carbon dioxide. If oxygen is present, it will accept oxygen. But always carbon dioxide को present होना है, because it has to accept this carbon dioxide and fix it in the form of a glucose molecule. But अगर दोनों में affinity देखें तो it has high affinity for carbon dioxide than oxygen. So it has only one side where oxygen and carbon dioxide binds. So this is again an important point. ऐसा नहीं है कि इसके पास दो साइड है. It is only having a single binding pocket in which either carbon dioxide or oxygen can bind, depending on the situation. And once this oxygen, it is inhibiting the process of photosynthesis. That is what called as Warburg effect. Now, what is this Warburg effect? Important. It is observed in the case of photorespiration, where photosynthesis is inhibited due to higher concentration of oxygen into the atmosphere. So we have already covered this diagram. Now let's move to the questions. So the organelles which take part in photorespiration are. Yes, Selva, we are back now. There was a technical glitch, so now it is sorted out. So I hope you can now see the slides, right? The organelles which can take part in photorespiration are, recall the code, Selva option 3. The code जो मैंने बताया for this photorespiration, it is PCM. But if you want to know the order, it is CPM. So anyways, you have to remember the organelles so you can remember physics, chemistry, maths. So where you are finding it? Peroxisome, chloroplast, as well as mitochondria. So the right answer is option number 3. So photorespiration is favored by. Next question. Very good, Srivani. Satyabrata. So photorespiration is favored by. It is low carbon dioxide as well as high oxygen, where RUBP, carboxylase oxygenase enzyme, it is acting as an oxygenase. So it is always high O2 as well as low CO2. लाइट का तो बात ही नहीं होता फोटोरेस्पिरेशन में, रॉन्ग. टेम्परेचर, लो टेम्परेचर नहीं. अगर यहाँ पे हाई लिखा होता तो ये भी ऑप्शन सही हो जाता, बिकॉज दिस O2 इज कॉजिंग दिस टेम्परेचर हाई. तो अगर यहाँ पे हाई O2 होता तो टेम्परेचर भी हाई होता. सो अगेन दिस ऑप्शन इज रॉन्ग. लो O2 एंड हाई CO2, रॉन्ग. लो CO2 but high O2. I hope this photorespiration is clear to everyone. Yes, very good, Srivani, Silva. You all are correct. So now we have covered all the pathways. We have seen dark reaction, C3 cycle, C4 pathway also we have seen, and we have also covered how RUBP, it is acting as an oxygenase. It is because of the high concentration of oxygen in the atmosphere that is hampering the C3 cycle to run. And why these pathways named as C4, C3, C2? Because of the stable product that is first forming in the reaction. So now we'll understand what are the factors that are affecting the process of photosynthesis. So there can be internal factors that are of the plant itself or can be external too. So what are the internal factors? The internal factors can be number, size of the chloroplast, mesophyll cells, orientation of the leaves, how chloroplasts are arranged in the leaves, how CO2 concentration, if it is there or not, what is the optimum range for CO2, and what is the amount of chlorophyll that is required for the process of photosynthesis. Or external factors, it can be sunlight जो कि बाहर से आ रही है, it can be water if we are giving water to the plants, if water is having optimum amount of plants is having optimum water availability, CO2 concentration if it is there in the atmosphere, as well as temperature. So light. So the first factor over here is light. Light के लिए कभी जंगल में देखा होगा देयर आर बिग प्लांट्स और यहाँ पे शेड अवॉइडेंस में कुछ छोटे छोटे प्लांट्स भी होते हैं ट्रॉपिकल फॉरेस्ट में दैट आर नॉट गेटिंग सफिशिएंट अमाउंट ऑफ लाइट सो दीज प्लांट्स आर नॉट गेटिंग सफिशिएंट अमाउंट ऑफ सनलाइट क्यों क्योंकि ये हमेशा शेड में रह जाते हैं सो फॉर दीज काइंड्स ऑफ प्लांट्स व्हिच आर अवॉइडिंग लाइट्स दे हैव अ लीनियर रिलेशनशिप इनका लाइट के साथ लीनियर ग्राफ जाता है बट प्लांट्स दैट आर गेटिंग लाइट्स रेगुलरली उनका ग्राफ ऐसे जाता है हाउ बिकॉज पहले तो ऑन इंक्रीजिंग द लाइट this photosynthesis is increasing. So we have light intensity on the x-axis and whereas photosynthesis, rate of photosynthesis on the y-axis. So first you see if we are increasing this light, so this photosynthesis is also increased. It has a linear pathway. But after some time, 10% saturation, this process is saturated. If you increase the light intensity also, this photosynthesis is not increased. That is, it is becoming constant. Why? Because it has no effect on the process of photosynthesis. So firstly, if light will increase, the photosynthesis will increase, but after some point of time, it is getting saturated. But it is not the same case in the case of chote plants that are avoiding light and who are suffering shade avoidance. For them, the graph always goes linearly. ठीक है so there is a really linear relationship between the incident light and CO2 fixation rate at low light intensity मतलब ऐसे प्लांट जिसको बहुत कम लाइट मिल रही है जो कि बड़े बड़े पेड़ से छुप जाते हैं at higher plant intensity gradually the rate does not show further increase as other factors become limiting बहुत सारे फैक्टर्स हैं जो कि एटमॉस्फेयर में खत्म हो जाते हैं जिसके कारण से लाइट अपना प्रॉपर फोटोसिंथेसिस रेट शो नहीं कर पाता so at some point of time it is getting saturated so the second factor over here is carbon dioxide so it is a major limiting factor because we know the carbon dioxide gas it is present in the atmosphere in only 0.03 to 0.04%. If we increase the carbon dioxide concentration in the atmosphere, it will cause damage to the plant. So it should always be between 0.03 to 0.04. एक नोट बना के रखना है the C4 plant they are saturating at 360 ppm or microliter or C3 plant they are getting saturated at 450 ppm because C4 plants में अवॉइड कर लिया है photorespiration phenomena को and that is why they are getting saturated at 360 but in C3 plants these plants are suffering from photorespiration का phenomena that is why they are saturating at 450 ppm of light. So make this a start point. For C3 it is 450 and for C4 it is 360. कैसे learn करना है? C3 में 3 is coming but 450 में 3 नहीं आ रहा है, तो उसको select करना है जिसमें 4 आ रहा है. But in C4 plant 360, तो 3 is coming but 4 is not there, so 360 ppm, that is parts per million. So the third factor over here is temperature. So light reactions are temperature sensitive and they are they are less affected. Whereas the C4 plants, they respond to higher temperature. So again, you can have a question from here. What is the optimum temperature for C4 plants? ताकि वो अपना C4 pathway run कर सके. It is 35 to 40 degree centigrade. But what in case of C3 plant? It is 25 to 30 degree centigrade. So C3 plant have a much lower temperature optimum, that is 25 to 30 degree in range. So now water. Now the fourth factor is water. We have seen light, carbon dioxide, temperature. Now the fourth factor is water. So if there would be a water stress, it will causes the stomata to close. And if stomata will be closed, no CO2 will be accepted. So it will definitely affect the rate of photosynthesis and it will reduce this photosynthesis, it will reduce. So water stress should not be there. If water will be there, it will open this stomata. Carbon dioxide concentration will be high because it is absorbing carbon dioxide from the atmosphere, and the photosynthesis rate will be more. And if it is not there, तो stomata will be closed, carbon dioxide accept नहीं होगा, rate will be affected. And अगर बहुत ज़्यादा water की कमी होगी, it will lead to the wilting of the leaves as well as the plants. So we have covered 4 factors: light, carbon dioxide, temperature as well as water, how these are affecting the rate of photosynthesis. So thank you everyone. We have completed this photosynthesis series. Yesterday we have covered the mechanism of photosynthesis, light reaction, and today we have dealt with the carbon fixation cycle, that is C3 cycles, and all the pathways that are taking place in photosynthesis. So thank you once again for watching this session. If you like the session, do not forget to like, share and subscribe. Also, after completing this session, you will get a feedback form. So please make sure to fill that feedback form, and you can also feel free to give the feedback for this session. So thank you once again. Wish you all the best. Meet you in the next video. Till then, bye-bye. Take care.